In a previous class, we have learned about digestion and absorption of protein. Now, today, we will learn about metabolism of amino acids. We will learn about generalized metabolism of all amino acids. So, as we know, metabolism means a combination of two words, that is anabolism and catabolism. So, the anabolic reactions, the proteins, they are synthesized from amino acid, that is called anabolic reactions, and it produces the few specialized product like creatinine, heme, purine and pyrimidine bases. Like catabolism, where the dietary proteins and the body proteins, they are broken down to individual amino acids. So this is metabolism is a general concept. It includes two things, anabolism and catabolism. So the protein undergoes, this amino acid undergoes certain anabolic reactions, as well as the protein that undergoes catabolic reactions. This general metabolism of all the amino acid, it includes transamination reaction, that ammonia formation and the detoxification of that ammonia and its excretion as a urea, synthesis of other non-essential amino acids, gluconeogenesis from amino acids. So this amino acid also helps in the synthesis of glucose, complete oxidation of amino acid, and other metabolic functions like conjugation, methylation, amidation, etc. So these are the different processes that comes under the metabolism of amino acids. The first and foremost step in the catabolism or the degradation of amino acid is to remove the amino group of amino acid as an ammonia. So this is the general structure of amino acid, that having amino group, carboxylic group are for the side chain. So the first step for the amino acid degradation is removal of this amino group as ammonia, and this is the structure of ammonia. The major source of ammonia is from the amino acid degradation, while the small quantity of ammonia that also comes from the catabolism means degradation of purine and the pyrimidine bases. Process of amino acid metabolism starts with the transamination reaction. The name itself suggests a transfer of amino acid. Transfer of amino group. Amino group from an amino acid transferred to a keto acid and resulting in formation of new keto acid and new amino acid. This reaction is called transamination. And for this reaction, what we required is PLP as a coenzyme. In almost all the cases, the amino group from an amino acid is accepted by a keto acid, that is alpha ketoglutaric acid, and by accepting this amino acid, this alpha ketoglutaric acid converted into newer amino acid, that is glutamic acid, and this is a reversible reaction. So almost all the amino acids metabolism starts with the transamination reaction. Here you can see transamination reaction in which glutamic acid and pyruvate, this is a keto acid, this is an amino acid, transaminated to alanine. This keto acid get converted into alanine, and this amino acid, that is glutamic acid, converted into alpha ketoglutaric acid. So this is a reversible reaction by which this also get converted into this and alanine get converted into pyruvate, and the enzyme is alanine amino transferase. So the amino group is interchange between the alanine and glutamic acid. And as I told you, in most of the cases, the amino group of an amino acid is accepted by alpha ketoglutarate acid and is get converted into glutamic acid. Now let's see the importance of the biological significance of transamination. The first of all, it is the first step of catabolism. As I told you, if any amino acid undergoes a metabolism, the transamination is the first step by which the ammonia is removed from the amino acid and the remaining carbon skeleton enters the catabolic pathway. The second, synthesis of non-essential amino acid. So almost all the non-essential amino acid can be synthesized by the body from corresponding keto acids by this reaction. And those amino acids which cannot be synthesized by this manner, this is called essential amino acid, then that we have to take from the diet. Like in previous example, we have seen that pyruvate get converted into alanine with the help of transamination reaction. So this alanine become non-essential amino acid. Here you can see alanine to pyruvate, okay, or oxaloacetate to aspartic acid, so or the alpha ketoglutarate to glutamic acid. So by this way we can synthesize non-essential amino acids. The third significance of this transamination reaction is interconversion of amino acids. This is helpful to equalize the quantity of two different amino acids, and this is also called equalization of quantities of non-essential amino acid. So this was the significance, and as I told you, for all the amino acid undergoes transamination before their metabolism, but there is an exception. This LTP, that is lysine, threonine and proline, they are not transaminated before their metabolism. So this is an exception. They follow the direct degradative pathway. So this is important for the MCQ point of view. Which amino acid does not undergo transamination reaction for their metabolism? So it may be lysine, threonine or proline. Now again we see the significance of the enzymes involved in transamination reaction. This AST and ALT, that is aspartate transaminase and alanine transaminase, they are induced by glucocorticoid and favors the gluconeogenesis. And these enzymes also AST, that is SGOT, increases in myocardial infarction and can be or was previously used as a cardiac biomarker. And this ALT and SGPT, also SGPT, that increases in the liver disease, that is in hepatitis. AST, that is also elevated in alcoholic liver disease. So this is the significance of transaminase enzymes. Then comes the second reaction, that is transdeamination. It's a coupled reaction. First transamination, that is transfer of amino group, then it is followed by oxidative deamination. This takes place in the cytoplasm of all the cells in the body. The amino group is transported to liver as a glutamic acid. As we have seen in most of the cases, alpha ketoglutamate acid that act as a acceptor of amino group and get converted into glutamic acid. So that amino group in the form of glutamic acid is transported to liver, where it will be oxidatively deaminated in mitochondria of the hepatocytes. The two components of the reactions, they are physically far away. The transamination reaction occurs somewhere else and also in the cytoplasm, while the oxidative deamination that takes place in the mitochondria of hepatocytes. So this reaction takes place at a two different sites, but it is physiologically coupled. Hence, it is called trans-deamination. Okay, let's see about that. This oxidative deamination of glutamic acid or the glutamate. So this glutamate comes from the transamination reaction. Then it transported to the liver, where in the liver it will be oxidatively deaminated. That means there will be removal of amino group with addition of oxygen with the help of glutamate dehydrogenase. There is, you can see here, this is a reduction, this is oxidation, and glutamic acid get converted into alpha ketoglutarate, and that ammonia that carried by the glutamic acid amino, it will be released here. This glutamate dehydrogenase is an allosteric enzyme. It is activated by GDP, ADP and NAD+, so it is activated from the substrate side, and it is inhibited by the product side, that is from the NADH, ATP and GTP. So here there is trans-deamination reaction. So any amino acid, this is the keto acid. There is a transamination. This amino acid converted into keto acid, and this alpha ketoglutarate keto acid converted into glutamic acid. So this occurs in the cytoplasm of all the tissues of body, and then this glutamic acid transported to the liver, where in the liver it undergoes oxidative deamination and it forms alpha ketoglutarate by the glutamate dehydrogenase. So this is trans-deamination reaction, coupled reaction in which there is a first transamination followed by oxidative deamination. Till now, what we have seen is the major pathway by which amino acid undergoes metabolism. There are other minor pathways for the deamination of amino acid. The first one, with the help of L-amino oxidase, it can act on all amino acids except hydroxy and dicarboxylic amino acid like serine, threonine, glutamic acid and aspartic acid. And this enzyme uses FMN as a coenzyme. And the peroxide formed that will be decomposed by catalase. Here you can see any L-amino acid that undergoes deamination then can convert it into amino acid and followed by keto acid, and in this reaction there is a production of hydrogen peroxide that will be taken care by catalase enzyme. Then D-amino oxidase, it can oxidize glycine and any D-amino acid that may be formed by the bacterial metabolism. As we already know that all the amino acid in our body, there are of L varieties, and this reaction uses FAD as a coenzyme. The next pathway is with the help of monoamino oxidase, that is MAO. Ammonia may be formed in the body through minor reactions like oxidation of monoamines by monoamino oxidase. Dehydratase that act on the mostly hydroxy amino acid to remove ammonia, like this is serine with a hydroxyl group. With the help of serine dehydratase, that hydroxyl group removed in the form of water converted into amino acid, and this amino group is removed and get converted into pyruvate. Same threonine get converted into alpha ketobutyric acid. Another pathway that is non-oxidative deamination, that is desulfhydrase, that causes deamination and trans-sulfation, in which cysteine get converted into pyruvate. there is a transamination reaction. Then histidase enzyme that converts histidine to urocanic acid. So the ammonia may also be produced in GI tract by bacterial putrefaction. So not only by the transdeamination, the small amount of ammonia is also produced by the gastrointestinal bacteria. Now, this whatever ammonia is produced by myogen pathway or major pathway or by the oxidative deamination, that is toxic, and excess quantity of this ammonia causes toxicity in the brain. So, the being highly toxic, this ammonia should be eliminated as and when it is formed. Even the small quantity produces the brain toxicity. So ammonia is produced by almost all the cells including neurons, and we have to take care to remove this ammonia. So the intracellular ammonia, whatever is produced by any reaction, that will be trapped by glutamic acid. And this glutamic acid will take that ammonia and get converted into glutamine in almost all the cells, but this is mainly occurs in the brain. So this becomes the first line defense. Any ammonia is produced in the cell, that ammonia will be taken up by the glutamic acid and get converted into glutamine. So this glutamate take up this ammonia, converted into glutamine with the help of glutamine synthetase enzyme. So this is first line defense. This ammonia is toxic, taken up by the glutamic acid, and itself get converted into glutamine. So this glutamine that is synthesized down from any part of the body almost through the brain will be transported to the liver, where this reaction is reversed by the glutaminase. So the amino group from the glutamine will be released in the form of ammonia by the group of glutaminase enzyme, and this glutamine get converted into glutamic acid. Okay, so the same way the glutamic acid helps in the transport of ammonia from brain or other part of body to the liver. The same way aspartic acid also get converted into aspargine, and again it comes to the liver and get converted into aspartame, and also helps in the transport of ammonia. So it is something like this. Glutamic acid take up this excess ammonia with the help of glutamine synthetase required ATP, and it forms a glutamine, and this glutamine transported to the liver, and in the liver with the help of glutaminase, this excess amino group will be released in the form of ammonia, and it gets converted into glutamic acid. So by this mechanism, the ammonia that produces in the brain or neuron or other cells of the body transported to the liver. So here you can see this from this glutamic acid breakdown, the deamination from the amino sugar, pyrimidine, putrefactions by the bacterial putrefactions, the glutamine and asparagine that brings the ammonia to the ammonia pool. This will be taken up by the glut and get converted into glutamine and asparagine, and the major portion of this ammonia is detoxified in the form of urea. Now, transport of ammonia inside the cell. The transamination produces the glutamic acid. We have seen that alpha ketoglutarate, that is a keto acid that accepts the ammonia and get converted into glutamic acid. Now the final deamination and the production of ammonia is taking place in the liver. So this glutamic acid acts as a link between amino group of amino acid and ammonia. So the amino group from any amino acid that will be transferred to alpha ketoglutarate acid, it forms a glutamic acid. This glutamic acid comes to the liver and releases that amino group in the form of ammonia. So because of this reason, the concentration of glutamic acid is 10 times more compared to any other amino acid. The glutamine is the transport form of ammonia from brain and intestine to the liver that we have already seen, while the alanine is the transport form from the muscles. So the amino group from the skeletal muscle will be transported in the form of alanine to the liver. So now the final disposal from all the parts or all the cells that ammonia comes to the liver from all over the body is detoxified to urea by the liver cells, the hepatic cells, and then this urea is excreted through the kidney. So the disposal of ammonia occurs in the form of urea. That urea is synthesized in the liver, and after synthesis that urea is excreted through the kidney. So the urea is the end product of protein metabolism. Okay, so since mammals including human being that excretes amino nitrogen mainly as urea, they are referred as a ureotelic. So this is all about general concept of amino acid metabolism. In the next lecture, we will go through the urea cycle, how this ammonia that reach to the liver and detoxified in the form of urea, that we'll learn in the next lecture. Thank you so much. Just can for the more update you follow channel that is on the Insta account that is @biochemistry for student.